Welcome back, students. So for this part of Chapter 10, we are going to be discussing the synapse and the synaptic transmission and how the action potential propagates or spreads along the axon. So first off, what is the synapse? Remember, the synapse is where a neuron communicates with either another neuron, as you can see here in this image, or it could also be the place where a neuron communicates with a muscle or a gland. So in this case, you see here two neurons connecting. So this is the axon of one and connecting to the dendrite of a second neuron. So this neuron before the synapse is known as the presynaptic neuron, and the neuron after the synapse is the postsynaptic neuron. So the impulse will be going from presynaptic neuron to the postsynaptic neuron. There is a gap, though, between them. So if you zoom in to the synapse, you can see here, this again is the terminal knob or the end of the presynaptic neuron. There's a little gap in the middle, and that is known as the synaptic cleft. And this right here is your postsynaptic neuron. So how is it that things are transmitted from pre to postsynaptic neuron? First off, let's take a look at the structure. This is a bigger picture of the synapse. Here again is your presynaptic neuron, the axon terminal, or the synaptic knob. You can see these secretory vesicles that are full of a neurotransmitter. And then there's a mitochondria. What happens is that when the action potential reaches the end of the neuron, that opens up calcium ion channels that are found on the cell membrane of the nerve. Calcium enters into the cytoplasm of the neuron. So that increases the intracytoplasmic calcium levels, which means in plain English that the levels of calcium inside of the neuron are increased. The calcium helps in the release, so you can see here these calcium ions help in the release of the neurotransmitter from the vesicle. The neurotransmitter pretty much swims across, diffuses across towards the postsynaptic neuron. In the postsynaptic neuron, you can see these purple receptors. When the neurotransmitter attaches to the receptors, activating them, now you have transmitted the action potential from the pre down to the postsynaptic neuron. Oh, something to keep in mind that when you activate the postsynaptic receptor, that does not necessarily mean that you have activated the nerve. Some of these neurotransmitters actually inhibit nerves. So these could lead to either excitatory or inhibitory effects on your postsynaptic neuron. Now, what is the cell membrane potential? So potential means electricity. And remember we said that there is a potential, or the cell membrane acts like a barrier between the inside of the cell and the outside of the cell. The inside of the cell is more negatively charged in relationship to the outside of the cell. And we call that, because we have a positive side and a negative side, we call that polarization. So across the cell membrane, there is a polarization where, again, intracellularly, inside of the cell is negative in relationship or in comparison to the outside of the cell. So again, inside of the membrane is negatively charged with respect to the outside of the membrane. And that is due to the unequal distribution of ions. So we have sodium, potassium, chloride, bicarbonate. There are also proteins that are involved. Some of these are positively charged. Others are negatively charged. They are not equally distributed, but the way they are actually distributed leads to that the inside of the cell is negative and the outside of the cell is positive. Again, just like a battery where we have a positive pole and a negative pole. That fact is extremely important for the propagation or the conduction of the impulse on the neuron, and it's also extremely important for muscle contraction. So what helps in the distribution of ions? We're going to be talking specifically about sodium and potassium. So remember we said that the ions are not equally distributed. The major cation found inside of the cell are potassium ions. Those again are your major intracellular positive ions. While your major extracellular, so outside of the cell, your major cation extracellularly are your sodium ions. But wait, I thought you just told us that the inside is negative and the outside is positive. Yes, but I also said that it is due to the unequal distribution of ions. So inside we have potassium that are positive, we also have proteins that are negative, and we have other ions that are in play. Okay, so the inside of the cell, although there's more potassium inside than there is outside, but your overall net result is that the inside is still negatively charged in comparison to the outside. Okay, that distribution, the fact that potassium is higher in while sodium is higher out of the cell, that distribution is created by this pump right here, the sodium-potassium pump. It is an active pump that needs ATP. Okay, it is going to play a very important role in maintaining the membrane potential. Know that the cell membrane has what are known as leaking channels. These leaking channels allow for ions to leak in and out of the cell. So these potassium and sodium keep on leaking across the cell membrane. What keeps them in check and keeps that distribution where potassium is always higher on the inside and sodium is always higher on the outside would be that sodium-potassium pump. Now, which one is more important in maintaining polarity across the cell membrane? That is actually potassium. So potassium plays a bigger role than sodium does in maintaining what is known as the resting membrane potential. So the sodium-potassium pump will always be transporting 3 sodium ions out of the cell and 2 potassium ions back into the cell. Okay, we have also these ion channels. They're usually closed. When they do open, they would allow for passage of ions. These channels are specific, meaning if this channel was for sodium, it would only allow the passage of sodium. Likewise, if the channel was specifically for potassium, it would only allow the passage of potassium. So these ion channels, they are closed. We need to open them up in order for an action potential to occur. To open them up, we have different keys. That key could be a neurotransmitter, so it could be a chemical. It could be maybe pressure, so that would be a mechanical opening or of the receptor or of this channels. Okay, so the gating, we call these gated channels, where the channels again are usually closed and we have to activate them. We have to open them up. And again, the key to open them up are very different from one receptor to another. So we have what are known as chemically gated channels or ligand gated channels. They're the same thing. And those are opened up with chemicals, neurotransmitters, while others are known as mechanically gated channels. And these open up with mechanical changes like pain, touch, pressure. We also have what are known as voltage gated channels. And these are activated when the membrane potential changes its voltage. Okay, so remember we said that inside is negative, outside is positive. If that changes, the change in voltage can lead to the activation of these voltage gated channels. Now, what is the resting membrane potential? That is pretty much what we have been talking about so far. It is the potential across the cell membrane at rest, where again, mainly potassium is on the inside, sodium is on the outside. The inside of the cell is negatively charged, and to be exact, it is at negative 70 millivolts. Okay, so remember that it is at negative, so you want to always remember that negative sign. Negative 70 millivolts, and again, due to the unequal charge distribution. So this is for a typical neuron at rest. Okay, remember we always have the sodium and potassium trying to leak through the cell membrane. The sodium-potassium pump just keeps on pumping them back where they belong. Now, in order to lead to an action potential, we have to activate certain gates. And we will do so, we'll explain this kind of like in a story kind of way. So let us take a look. Here we're also looking at the resting membrane potential. So there again is your inside of the cell with your higher potassium levels. And this is your outside of the cell with your higher sodium levels. Remember again, the inside at rest is negative 70 millivolts. There's always leaking of potassium out of the cell, and sodium is always trying to leak into the cell. Again, everything goes back to normal through the function of the sodium-potassium pump. Okay, now what is a local potential change? Neurons are excitable nerves, meaning that neurons are made to conduct electricity. So they do have the potential to make their own electricity. Okay, they can detect different changes, different stimuli, whether they're visual or auditory stimuli, and they can respond to that in the form of changing their resting membrane potential. They would open certain gates. When you open different gates, that allows for ions to go either in or out, depending on which gate you opened, and that will change the membrane potential. And I am going to go to the image to explain different things here. So your resting membrane potential is at negative 70 millivolts. When you activate the nerve, the very first thing in nerve activation, you either activate it due to a mechanical stimulus, like pain, touch, or pressure, or it is activated by a chemical, like a neurotransmitter. And that would open up either a chemically gated sodium channel or a mechanically gated sodium channel. But in both cases, you've opened up sodium channels. Now remember that sodium is higher on the outside. So when you open up sodium channels, sodium ions are going to go into the cell. That will change the potential from negative 70 going towards the zero. Okay, so you can go all the way to negative 55. If we reach that negative 55 millivolts, and that is known as your threshold potential, that change in voltage from negative 70 to negative 55 opens up a second kind of channels, and those are known as voltage-gated sodium channels. So far, we only opened up sodium channels. Your very first one are either mechanically or chemically gated, and that leads to that initial change in the membrane potential from negative 70 to negative 55. If the stimulus was strong enough to reach that threshold potential, then we are going to open up a second kind of sodium ion channels, and those are the voltage-gated sodium channels. Sodium then is going to rush. There will be a rush of sodium into the cell, and that leads to what is known as depolarization. So in depolarization, we opened up sodium channels. The voltage now goes into the positive territory, so we can reach up to positive 30 or positive 35. We'll stick to what's in your textbook of positive 30. And then at that point, at positive 30, the sodium channels are deactivated, and we open up a third kind of channels, and those are your potassium channels. Remember, potassium concentration is higher inside than out. So potassium will rush out of the cell. Potassium ions that are positive are now rushing out of the cell. That would lead to repolarization. We are losing a lot of these positive ions. So we go back to becoming negative, but potassium ion channels are very slow in shutting down. So not only do they go to -70, but these cells get hyperpolarized where they could go to -80, even all the way down to -90 millivolts. And that is called hyperpolarization. Again, it is due to the slow shutting down of your potassium ion channels. Now, at -80 or -90, the potassium channels close, and then the cell has to readjust back to the resting membrane potential. And that correction, or going back to our resting membrane potential, is done through the sodium-potassium pump. That is the function of the sodium-potassium pump. So to break this down again, okay, your very first part here, when you stimulate the nerve, you are opening up either mechanically gated or chemically gated sodium channels. Sodium goes into the cell. If the stimulus was strong enough and you opened up enough mechanically or chemically gated sodium channels, and enough sodium goes in, that changes the potential to -55. That is your threshold. At that point, you're going to open voltage-gated sodium channels. Sodium rushes in, leading to depolarization. When we reach +30, sodium channels are deactivated, and we open up the potassium channels. Potassium rushes out, escapes from the cell, and that because the potassium ion channels close very slowly, they do lead to hyperpolarization. At -80 or -90, the potassium ion channels close, and then the nerve restores back to its resting membrane potential by the action of the sodium-potassium pump. Again, pumping 3 sodium ions out and pumping 2 potassium ions back in. So that is what an action potential looks like, and this is what we were talking about. So again, when you activate, in this case, we are using neurotransmitters, so we are going to activate these ligand or chemically gated sodium channels. That leads to some change in the potential. If it did not reach a threshold, you see here it goes back to normal. These are known as local potential changes or graded potentials. But if the stimulus was strong enough and we opened up enough of these chemically gated sodium channels where lots of sodium came in, enough to get us up to this threshold stimulus of -55, that will lead to the opening or the activation of your second kind of channels. Those are known as voltage-gated sodium channels. Again, they were open due to the change in voltage from -70 to -55. That leads to the upstroke, or the process of depolarization, up until we reach +30. At that point, at +30, potassium ions open, sodium ions deactivate, potassium is going to escape from the cell. They shut down very slow, and that leads to the hyperpolarization. At -80 or at -90, this potassium channel shut down, and then we restore the resting membrane potential due to the sodium-potassium pump. You can see here the ion movement during an action potential, that the action potential goes in the direction away from the axon cell body. So had this been the cell body, the action potential will be going down in this direction towards the end or towards the axon terminal. Now, all of the changes that we talked about, we start off, the voltage-gated channels are found in the axon hillock, and that is why it's known as the trigger zone, because if these channels are triggered to open, these voltage-gated sodium channels were triggered to open, this is when we are going to actually produce an action potential. Remember, you have to reach that threshold of negative 55. If you do not, again, everything goes back to normal. It's a sub-threshold stimulus. So going back to this image here, sub-threshold stimulus leading to a sub-threshold response. Nothing is recorded. Nothing is going to happen to that nerve. These are known as local potential changes. They are also known as graded potential changes. Again, just to kind of wrap up a couple of things, what causes depolarization, and that is the opening of your voltage-gated sodium channels. So sodium rushes into the cell. What leads to repolarization? That is the opening of the potassium ion channels, and potassium rushes out or escapes out of the cell. That temporary hyperpolarization is due to the fact that potassium channels close really slow, and more potassium than needed actually escapes the cell. How does the cell reestablish your resting potential? That would be due to the action of the sodium-potassium pump. Now again, for the conduction of the nerve impulse, it'll be along the axon, away from the cell body, and the direction of conduction is always in one direction, from cell body towards the axon terminal, and never in the opposite direction. So these are the events that lead to the impulse conduction, and this is exactly what we were talking about, so you can pause the video at this point and kind of read through this. Now, what is the all or none response? Remember, we said that a local potential is a graded potential. It's that happened due to a subthreshold stimulus. If we do not reach the thresholds of negative 55, nothing happens, everything goes back to normal. Well, that's different than an actually action potential. So when we do reach the threshold, an action potential occurs, and that is an all or non response. Meaning, and I'm going to go back to that image, as long as we are subthreshold, changes are just happening here locally and nothing propagates along the nerve. But when we do reach that threshold, we are going to make this action potential in its entirety. Okay, so it's either an this one is the all or none kind of thing. Okay, and that's what the all or none means when we're talking about an action potential. So you either get the whole response or you get none of it. Okay, so regardless of the strength of the stimulus. So let's say you get, let's say you hurt yourself, and then on a scale from 1 to 10, you grade it as a 2. Okay, let's say you hurt yourself again, and then on a scale from 1 to 10, now you grade it as a 10. That is not going to change the shape of this curve. So that action potential curve will be the same regardless of how strong the stimulus is. So how is it that we interpret these different stimuli or the strengths of the stimuli? That would be due to the frequency of the action potential. So not the amplitude. The amplitude remains exactly the same, whether your pain is at a 2 or pain is at a 10. But it is all in the frequency. So how many impulses per second are being propagated on that nerve? At a 2, the frequency is lower, while at a 10 level of pain, your frequency is much higher. So again, your all or non response, it either happens or it does not happen. You either feel the pain or you do not feel the pain. When you go through the all response and an action potential actually does occur, again, the amplitude of the curve is the same regardless of the stimulus. We interpret the different intensities of these stimuli due to the change in the frequency of the action potential, not the amplitude. Now, what is the refractory period? Remember we said that the action potential goes from the cell body down the axon, away from the cell body towards the axon terminal. And that is due to the refractory period. Refractory means stubborn. And there are two different kinds of stubbornness that the nerve goes through. Generally speaking, refractory period means that when changes occur when these electrical changes happen, sometimes the nerves are re-stimulated. Okay, will the nerve respond to that second stimulus or not? Well, that depends. Okay, sometimes we have what is known as an absolute refractory period, where no matter how strong that second stimulus would be, the nerve is never going to respond to that. And that would be due to sodium channels are already activated. Okay, remember, in order to feel something or to have an action potential, it all really depends on the opening of sodium channels. Well, what if the sodium channels are already all open? So how are you going to respond to that second stimulus? So your absolute refractory period is usually during the period of depolarization where your sodium channels are already open. That is an absolute refractory period. No matter how strong that second stimulus is, the nerve is stubborn, all of its sodium channels are open, it's not responding. A relative refractory period, that occurs during the process of repolarization. Now the sodium channels, some of them are able to respond. Okay, so if that second stimulus came and it was stronger than the first one, now the nerve is able to respond to it. Okay, so relative refractory period is time when only a high intensity stimulus can generate another action potential. And this usually happens during the process of repolarization, where some sodium ion channels are available to reopen. And in that case, what would happen is that another action potential will occur. Okay, so again, absolute, nothing is going to happen. Absolute stubbornness. Relative stubbornness, not as much. Okay, sodium ion channels are able to open if a higher than normal stimulus the nerve is exposed to a higher than normal stimulus. And that is why in impulse conduction, there is, and I'll go back to that in a minute. So in impulse conduction, the going from axon or from the cell body towards the axon terminal, that is why not a good image to use. Hold on. This one is better. So this is where the potential is going to happen. It'll propagate away from the cell body. And again, because it only has that one, it can only go in that one direction. You see here now the changes have propagated to the middle portion, where it would have one out of two options. It either goes backwards or forwards. If it went backwards, well, that cannot happen because this portion is in its absolute refractory period. So it cannot respond to anything. So really, the action potential only has one way to go, and that would be down this direction. So the absolute refractory period helps in making sure that the action potential goes down the axon in only one direction. Myelination definitely helps that process, because now instead of the action potential having to literally walk down the axon, it could jump down the axon. It's kind of like skipping. Remember we said that that myelin sheath is really made out of lipids. It's an electrical insulator. And because of these gaps, or known as nodes of Ranvier, it can lead to saltatory conduction, where the electrical changes, instead of having to go through the axon one step at a time, it could actually jump in between these nodes of Ranvier, and that, kind of imagine yourself walking as opposed to skipping. Which one is faster? So the skipping is obviously faster, and that kind of skipping, the Ranvier was French, okay, and he explained the process and in French, and so saltatory conduction, it really comes from the word skipping in French. Now for a little clinical application, so what would happen if there are changes in the ion concentration, whether in the extracellular fluid or, on the inside of the cell? So potassium, remember, potassium is really the master of all of this. So what happens if you have increase of potassium concentration in the extracellular fluids, known as hyperkalemia? Okay, that is going to lead to a change where the threshold potential is reached with stimuli of lower intensity. Okay, meaning that now the neurons are hyperexcitable. Okay, they're super sensitive, and that could lead to convulsions. What about if there is a decrease in the concentration of potassium in the extracellular fluid? So the person has hypokalemia. Now the neurons can be hyperpolarized, maybe meaning that we have pretty much numbed the nerves. It is very hard to generate an action potential, and that can lead to muscle paralysis. And that is why potassium levels are extremely important to maintain. Something to keep in mind, especially if somebody is taking hypertension medicine or diuretics in particular. Some diuretics get rid of potassium as well in urine, and that change or the disturbances of potassium levels can be dangerous to the patient. So they always want to monitor their potassium levels. There are what are known as potassium sparing diuretics. So they will still act as a diuretic, but keep the potassium in the body, hence the name potassium sparing. These are kind of diuretics they want to give to patients if they do experience disturbances of potassium ion levels when taking diuretics. Okay, a decrease in permeability to potassium ions. This is really what an anesthetic drug can do. Okay, it could use, that fact, the importance of these potassium ions in order to numb a neuron. Okay, so it'll stop the impulses from passing through around the axon. The impulses are not going to reach the brain, and therefore there is no perception and there is no feeling of touch or pain. Now, for the synaptic transmission, remember, this is all from nerve to something else. Okay, the transmission, in this case, we're really talking about nerve to nerve. So the transmission of a nerve impulse from one neuron to another is going to be dependent on a neurotransmitter. Neurotransmitter will have to diffuse across the synaptic cleft and attach to the receptor in the postsynaptic neuron. That's going to lead to activation of ion channels, sodium, as we were talking about in this case, if it is going to excite the postsynaptic neuron. If it's going to inhibit the postsynaptic neuron, then we could open up potassium channels or chloride channels. Okay, so it all depends on what the effect is going to be. If we want to excite the postsynaptic neuron, we are going to open up sodium ion channels to produce an action potential. But if you want to inhibit the postsynaptic neuron, we could do that by opening up potassium or chloride ion channels. Remember that chloride ions are negatively charged. So we have two different kinds of synaptic potential. There are EPSPs, or excitatory postsynaptic potential, but nobody has time to say that, so they're called EPSPs. Again, you're opening up, in this case, sodium ions. This leads to some depolarization. And if you reach the threshold potential, that would lead to an action potential. So an action potential can occur. It is more likely to occur or can occur in EPSPs. But if you open up potassium channels or chloride channels, where now you are hyperpolarizing the postsynaptic neuron, these are known as inhibitory postsynaptic potentials, or IPSPs. In this case, an action potential is definitely not going to occur in the postsynaptic neuron. What really happens is that you can see this poor nerve is getting impulses from several surrounding nerves. Some of them are EPSPs, some of them are IPSPs. So what is this poor nerve to do? It does what is known as summation. It'll sum up how many EPSPs am I getting versus how many IPSPs am I getting, and add those up and see, am I going to eventually be stimulated or inhibited? Okay, so it kind of like goes through its pros and cons and puts them together. That process of summation occurs right here in this funnel-shaped area known as the axon hillock, and hence that's why we call it the trigger zone. Again, is this axon or is this neuron going to be stimulated or inhibited? It's all about the summation process. This is actually what your nerve cell, your nervous system looks like. Okay, took away all of the other tissue. There's surrounding tissue, and that's what it looks like. I thought it looked pretty impressive and thought I'd share this with you guys. At this point, this really ends this portion, and I will see you in next episode.